Walk down any grocery aisle, and you'll see labels suggesting that yogurt, kimchi, or kombucha provide your daily dose of probiotics. Most people assume these everyday fermented foods are guaranteed to be probiotics. Science, however, uses strict standards that classify them entirely differently. Fermentation is a natural process of microbial transformation. It begins when naturally occurring bacteria or yeast feed on the sugars present inside raw ingredients, reshaping the chemistry, flavor, and texture of the food. Foods created this way are best understood as cultivated microbial environments. They hold broad, diverse communities of microbes. Because these are natural ecosystems, the exact strains and the total number of microbes present fluctuate wildly from one batch to the next. Researchers cannot guarantee which specific microbes are in every jar, nor can they promise those microbes will survive the journey through your digestive tract. Without that biological certainty, these broad environments don't meet the medical classification for true probiotics. During this transformation, a specific group called lactic acid bacteria goes to work. They convert natural sugars into lactic acid, which rapidly lowers the food's pH. This acidic environment acts as a natural preservative, safely extending the food's shelf life while blocking harmful organisms from taking root. This diagram shows exactly how fermentation alters nutritional chemistry. Many raw plant foods contain compounds that bind tightly to essential minerals, like zinc, calcium, and iron. When locked in these clusters, those minerals are incredibly difficult for the human body to absorb. As bacteria grow and process the food, they release microbial enzymes. These enzymes dismantle the molecular structure of the binding compounds, finally releasing the trapped nutrients. The primary biological value of fermentation lies in this chemical transformation of the food itself. It improves nutrient availability and makes the raw ingredients far easier for the human body to digest, independent of whether any live bacteria actually make it to your gut. In fact, a vast majority of the live microbes present in fermented foods die upon entering the highly caustic environment of the human digestive tract. Yet dead microbes still offer distinct biological benefits. When they expire, they leave behind what scientists call postbiotics. These are the physical leftovers, cell fragments, specific metabolites, and structural components that remain after a microbe completes its life cycle. These postbiotic fragments travel through the intestines, where they continue to interact with human cells. They provide chemical signals that influence immune system responses and help reinforce the structural integrity of the gut barrier. Fermented foods deliver immense health value through this combination of unlocked, easy-to-absorb nutrients and residual postbiotic fragments. Providing broad ecosystem nourishment does not require living microbes to be effective. True probiotics sit on the opposite end of the scientific spectrum from these broad, unquantified ecosystems. A probiotic is a specifically selected, highly vetted microbial strain, chosen because it performs a precise, clinically verified biological function inside the body. Many of these targeted health effects, like reducing specific digestive symptoms or actively fighting off harmful microbes, only trigger at precise, consistent doses. This requires a completely different standard of measurement than natural fermentation can provide. This split-screen chart illustrates that difference. Scientists count probiotics using colony-forming units, or CFU. This is a strict measurement of the exact number of living, functional microbes in a single serving, ensuring absolute consistency that natural batches cannot match. The title of probiotic is a clinical designation. It demands guaranteed, standardized numbers of living organisms that possess the specific biological tools necessary to execute targeted tasks. The human digestive system is an intentionally hostile environment. It is structurally designed to destroy unadapted organisms before they can reach the lower gut. In this cross-section of the upper digestive tract, we see the first major hurdle, stomach acid. Ordinary bacteria melt away here, but specifically selected probiotic strains are clinically screened to withstand these extreme pH levels and remain entirely intact. The second hurdle waits in the small intestine. Here, the body releases bile salts to digest fats. These salts are highly toxic to bacterial cell membranes and threaten to tear the surviving microbes apart. To counter this, specialized probiotic strains produce an enzyme called bile salt hydrolase, or BSH. This enzyme actively neutralizes the toxicity of the bile, allowing the microbe to pass safely through the chemical wash. True probiotics are defined by this exact evolutionary resilience. They possess the required biological tools to survive a digestive gauntlet that destroys nearly everything else. Reaching the large intestine alive is only part of the battle. To function, these microbes must physically anchor themselves to the intestinal lining. Otherwise, the body's natural processes will simply wash them out. Many effective strains produce exopolysaccharides, or EPS. These are thick, complex sugar coatings that the bacteria extrude to form a physical microbial shield. These EPS structures serve multiple purposes. They allow the bacteria to tolerate environmental stress, stick firmly to the slippery walls of the gut, and establish stable, temporary neighborhoods among the existing flora. Without these highly specific adhesion traits, even the hardiest surviving microbes would pass completely through the digestive tract without delivering their intended clinical benefits. The gut is a highly competitive, resource-scarce ecosystem. Even the most resilient, perfectly engineered probiotic strains will quickly fail to function if they are starved of nutrients. This is where prebiotics become critical. Prebiotics are specialized, indigestible plant fibers that humans cannot break down, but which serve as the optimal fuel source for beneficial gut microbes. When microbes ferment these prebiotic fibers, they generate short-chain fatty acids. These newly created compounds act as a major energy source for the colon wall, directly maintaining the structural strength of the intestinal barrier. Consuming live probiotics alone is insufficient. Their survival, growth, and biological output are directly dictated by the structural prebiotic fuel you provide to the receiving ecosystem. Fermented foods and targeted probiotics ultimately serve entirely different biological roles. Fermented foods provide broad ecosystem nourishment. They deliver unlocked nutrients, organic acids, and postbiotics that improve the baseline conditions of the gut. Targeted probiotics deliver clinical precision. They introduce precisely measured, heavily tested strains designed to execute specific biological functions that natural foods cannot guarantee. These two systems do not compete. They operate synergistically. Fermented foods prepare and maintain a supportive ecosystem, while targeted probiotics enter that prepared environment to provide specialized interventions. Distinguishing between broad microbial cultivation and clinical precision provides a clear roadmap for supporting the gut. It allows for a strategy that pairs the broad ecosystem support of fermentation with the targeted biological functions of evidence-based probiotics.